Hi, I'm Amanda. I'm your TA, and I am jumping on here to introduce myself, but also to share how I'm going to process the samples that some of our students dropped off. So, the first samples that were dropped off are from Jeffrey and Julian. Awesome. They're collected from cool places. So Jeffrey collected his from Jeffrey collected theirs from an aquarium filter, and Julian collected theirs from the bird rock boulders. So we'll see what kind of microbes grow on there. And importantly, we're going to do two different ways of getting the bacteria to grow on media onto bacterial agar plates, or to hard media. And so a plate, the solid agar plate looks like this. It's in a petri dish, and and then if you look at it from the side, you can see that there's about 25 mils of media that was poured into it and then allowed to cool so that it solidifies. So it's very similar to gelatin. The media itself is called Marine Agar. It's commercial. You can buy it from the store. If you look up BD or DIFCO Marine Broth 2216, you would find it online very easily. In it, it lists all of the ingredients, which are seen here. It's supposed to mimic the ingredients that are necessary, that are provided by the ocean naturally, a lot of the minerals and then also the metals. It's really easy to make media, especially when it's commercial like this, because it has instructions. So this one is going to say, measure 37.4 grams of this media in 1 liter of water, and then mix it and autoclave it, and you're pretty much ready to go. So now I've made my media a while ago, and so I have the solid plates. And the first question is how to go from a sample onto the plate. And it ultimately comes down to how many bacteria we would imagine would be on a particular surface and how many of those would be culturable. Because ideally what we're looking for is to be able to isolate a single colony. So you want it to be diluted enough that you can pick out one single colony and not touch any of the other colonies there. So we did two different approaches, and I'm going to show you what the differences are between them. We'll start with Julian's first, because he gave us a cotton swab. And in trying to get the bacteria off the cotton swab, I want to do a type of plating called spread plating. And spread plating resuspends all of the bacteria into a liquid. In my case, I'm going to be using filtered seawater. And then I will plate liquid, so that filtered seawater plus the bacteria, onto the plate, and then I'll spread it around evenly, so hopefully we get a bunch of single colonies. Now, in order to get it from the cotton swab, which is now in this 1.5 mil Eppendorf tube, what I did is I broke off the wood, and I put in 500 microliters of filtered seawater, and then I vortexed it. So it's a shaker machine that shakes very violently for 1 minute. And in doing that, we're hoping that a lot of the bacteria that were on the surface of the cotton swab were maybe dislodged, maybe that they're free swimming now in the liquid portion of this. So the first thing that I want to do is I want to take a sample of the full concentration from the cotton swab. So if I want to, first, when we're doing this kind of work, we're going to use sterile techniques. So we're going to use the Bunsen burner flame. In order to do that, we're going to turn on the gas, which is the lever right here. Might be hidden by some stuff right now, but I'm going to pull this lever, and you can hear the gas is now going. Now I'm going to use this lighter to light the flame, and I can smell the gas. And if you can't get this to go on the first time, then you want to turn off the gas, let it dissipate, and then turn it back on. So the first thing that we're going to do is we're going to take 100 microliters. It's already on 100, lucky day. 100 microliters of the full concentration. So I labeled the tube, and it just says JC full. And I'm going to use the pipette to pull up 100 microliters. And what I really want to do is I want to pipette mix really quickly. So in order to do that, I'm just going up and down with the plunger multiple times. Really want to mix it up in here and get any of the microbes that we have. And I'm going to pull up 100 microliters. And then I have plates that are already labeled here. So JC full, I labeled. So normally you want to be like by the flame. I'm going to lift up the side of the plate and just pipette the liquid onto the top. And so here, and I got rid of the pipette tip there. So here we just want to delicately put the liquid on the top. We're not trying to jam it in there. We just want it to rest on the top, okay? So from here, we're going to use these beads. They're called rattler beads. We call them spread beads. They're really just like mini marbles almost. And they're autoclaved, so they've been sterilized. And we're going to pass this through the flame. And now we're going to let out, maybe 10 of these beads onto the plate like that. There you go. We're going to pass this back through the flame so that we can be sterile. And now we're going to do, we're going to rattle them. So we're going to move them all across the surface of the media so that it distributes the liquid all around. So we want the liquid to be fully coating the top of the media. Where they get their names from, because they call them rattler beads. Okay, and then once we think that it's all fully around there, a couple more times for good luck, then we just get rid of the beads into an empty jar. I just have a jar that's for used beads. And I just tap it, make sure all the beads are out, and then this is ready to go in the incubator. So this is a full concentration version. And what I'm guessing is that we may get a lot of bacteria here, and maybe we get so many that we actually can't count them or select any because they're all touching their neighbors. And if that's the case, we want to make a diluted version as well. So the diluted version, we don't need much. We only need 100 microliters of it. But in order to make like an easy dilution, I'm going to do it in 500 microliters. And in order to do that, I'm going to take up 450 microliters of water, of the filtered seawater, which I have just in a tube nearby. And add it to a new tube. And that new tube is labeled JC 1 to 10. That's a 1 to 10 dilution. So I'm adding the water first into the tube, get rid of the tip. And then I'm going to add 50 microliters of the full amount. So back in our full tube, we're going to take 50 microliters of this, mix it again, and now we're going to mix it into the 1 to 10 tube. And here you want to do another round of pipette mixing just to make sure that it's fully all around. All right, and now we're going to take 100 microliters again. So we're going to change it from 50 to 100 and plate the diluted version, 100 microliters of the diluted version. Onto a new plate that is labeled 1 to 10. So here we have the name of the place, the initials of the student that collected it, the date that I plated it, and then what kind of concentration is on the plate. Again, we're going to pipette onto the surface of the plate. We're going to throw a few of the beads in there. Rattle them around. That's it. So we'll see what grows, Julian. And then for Jeffrey's is interesting because it's dark and it has sediment in the bottom. If you don't shake it up, it kind of looks like that. I expect there's gonna be a lot of crap in there. And so instead of doing a full version of this, we're only gonna do diluted versions. So just like we did the dilution for the 1 to 10 for Julian, we're gonna do multiple dilutions for Jeffrey just to make sure we can get individual microbes. And instead of just doing 1 to 10, we're gonna do 1 to 10, 1 to 100, and 1 to 1,000, just so that we can make sure we can actually get some good microbes in there. And the way that you can get to 100 or 1,000 really easily is you can do something called the serial dilution. So instead of trying to pipette a super small amount of the liquid, instead what you can do, and a super small amount of the liquid one time, instead what you can do is the same kind of dilution that we did for Julian, but then once you make the 1 to 10, you do the same exact dilution again to make a 1 to 100, and the same dilution again to make a 1 to 1,000. And we call this a serial dilution. Now, I'm not going to do this again the same way, mostly just because it's going to be the same process. But now you kind of know what the different techniques are and how you could go straight from a Q-tip or even straight from liquid onto a plate and then serial dilute and spread plate so that it is ready to go in the incubator and grow overnight. Cool. We'll see how this turns out. I hope we get some cool bugs.